Hello, welcome to MACSMIN 2024 again. Now, Olga Anosova from our group will talk about geographic-style invariant coordinates of protein backbones. Please, Olga.
Okay, thank you. Can you Great. Okay, thank you very much, Vitaly, and thank you for the opportunity to present work here. So my talk is about the geographic-style invariant coordinates of protein backbones. And despite having real experts on proteins in this whole and in this role, I hope it still be a novel talk and novel information for everybody, because I will be looking at the things obviously from a mathematical point of view or data science point of view. So data science starts with, well, data. So what kind of data we are talking about? The largest collection of data, as John mentioned in the previous talk, is the protein data bank, which is considered as a gold standard collection of experimental proteins, and that are written in entries coded by 4 symbols for each entry. And here are 3 examples for each of those 3 codes. So basically it's something like that. So that is a slightly unusual structures for my today's presentations, because it's actually viruses. But yeah, just to give the impression how complicated geometrically in shapes and overall they can be. And the middle one probably resembles the very commonly and sadly known shape nowadays since several years ago. So each entry in PDB can have a few versions models and consists of several entities, and if within each entity there are 1 or multiple chains also given by some coded IDs. So that's the structure of the data overall briefly. Now, the main thing in this data maybe is the sequence itself. So what is the sequence? The primary sequence, which also defines the primary structure of a protein, is just a linear chain of amino acids. And the conjecture is that this primary sequence of amino acids, this linear chain, uniquely defines the geometric shape or so-called tertiary structure in R3. Well, basically a very quick tutorial for the people who do not usually deal with proteins. So we start with some kind of basic atoms that form amino acids. And the basic ones are here, which is carbon, oxygen, H and N. And then they are combined into amino acids. So I think this one is I don't remember which one is glycine, how you pronounce it. I can write it down like GLY, never pronounced before. Yeah, so then they are combined with each other to form a longer sequences. So how they are combined is quickly depicted on this. Yeah, you can see my moving slider. They are combined like that. So firstly, there are 2 different carbons, and one is called alpha carbon because it has a specific role. It is connected to the side chains, which sort of define the variability of what we are dealing with. And the main structure that is always present there is the nitrogen carbon, alpha carbon, another carbon. And again, it is connected then to the same nitrogen, alpha carbon and carbon. And since alpha carbons are so important, sometimes only they are considered, just remove everything else. So sometimes only alpha carbons are considered at all. And from primary sequences, the main question is it possible to easily define, justify the 3D shapes? Okay, slightly closer and a bit more realistic look on the same construction. So firstly, whilst there are many amino acids in a protein, only 22 are involved in building a protein, well, and 20 major ones, that's what we routinely consider. We also denote, which is probably unusual again for biological people, we denote the alpha carbon atoms as A instead of Ca, just to save on basically pronouncing alpha carbon, yeah, and also in notations. So, and as mentioned, the protein backbone then can be represented as just the sequence of those major triplets of atoms, that is nitrogen, alpha carbon, and another carbon, so NAC. And then when we combine amino acids, we combine the so-called residues, and by the amount of amino acids we have, that is the number of residues in the full structure. So as mentioned, if we have amino acids, then they have the chain of length M, and this NAC structure of repeated 3 main atoms can be called as a backbone. Well, quite often backbone also oxygen is involved, but yeah, since the sort of primary line is traceable from N, A, and C, that's what we will call a backbone here. And yeah, as mentioned, alpha carbons also are important, and sometimes they are called just backbone trays. So sort of from the smallest bone to more bony structure and to the full protein structure itself. So the key question that this group is dealing with, being it proteins, crystals, or whatever, well, that's the also common question in mathematics. The key question is same or different, and how different. And we are dealing with mostly backbones today. So applied to the backbones, the answer is sort of obvious. Backbones for chains are the same if they have the same primary sequence. Yeah, we have the same primary sequence, which means that surely bonds are also the same and chains are the same, right? But we are mathematicians, and we know what mathematics is, which means that, yeah, let's try to answer this question again. Can we answer it differently? Well, yeah, we can. Another answer could be that backbones are considered to be the same if the number of the residues or the length is the same. But this equivalence is also possible. It's interesting to consider sometimes the chains of the same length, but obviously this equivalence or comparison type is weaker than by primary sequence, simply because there are many primary sequences with the same length. So it's possible, but it's just can be interesting. It's just weaker. Okay, so we are dealing with geometric data science, which means that we can also consider another possible answer. That is looking at the geometrical structure and shape. And we can say that if backbones are considered as ordered set of atoms, then they should have also the same sequence. Do you think it's a correct statement, Daniel?
I'm sure there are exceptions. So
Okay. But also another statement is that same sequences of proteins can have different backbone structures. Well, do you agree with that? That's yeah, less doubt on that. But the question is again, what do we mean by different? Yeah. So let's try to quantify that, because again, why is it important? Because we as trained humans can deal with that, but now we are more and more interested in applying machine methods to everything. And machines are very quick, but less trained. Which means that very precise definitions and ways to measure this similarity and difference can be indeed essential more. And what we consider to start with is the difference or similarity compared by rigid motion. Why we thought that it's meaningful? Well, because of the hypothesis that suggests that enzymes interact, well, basically based by shape. And by shape, I mean here that if we lock them or move them rigidly together, then they can fit as a lock and key mechanism. Well, that's again something that we've learned quite recently in COVID years. So it seems to be reasonable to look at rigid motion. So mathematically, any rigid motion can be considered as composition of translation and rotations. And it preserves the shape of the whole structure of the backbone. And the suggestion was that more flexible deformations can change its properties, which means that, yes, it is interesting to see how more flexible deformations behave, and I will get into it. But the simplest, strongest thing to actually consider is this rigid form, rigid shape, and rigid motion. So rigid motion in our case is considered to be the strongest equivalence on protein backbones. We consider the backbones by rigid motion. But as mentioned, don't worry too much. We realize that proteins are very flexible shapes, and I'll come to that as well. Okay, so we've considered several equivalences already when answering our question, same or difference. So we've started with the weakest one, which is length, that can be extended to sequences which will, even for the fixed length, form a set of much larger possibilities. But then, if we want to consider even further into the shapes, then the range of possibilities becomes actually infinite, even if we fix just one length. Okay. So how can we actually distinguish between all infinite many rigid classes? And yeah, since I'm doing the first talk in our group, I will quickly remind the very common notion in this group of an invariant. So invariant is just a property that is, well, invariant. That is preserved under something. And since we are dealing with equivalences, this property is preserved under the equivalence that we consider. In our case, it should be preserved under rigid motion. In this particular example, this particular case of proteins or backbones, if proteins are equivalent under rigid motion, then invariant should be the same. And again, simple mathematical thing to pay attention to is if-then logical construction. In mathematics, we all know that if-then does not routinely mean the converse. So if logical implication works in one direction, can we always state that it will work in another direction? Well, not really, which means that if invariants are the same, it's not guaranteed that backbones will be the same. So a very simple, again, analog of what is example of what is an invariant. Invariant can be, for example, the height of a person. If two people are not two people, but one person, surely the height will be the same. Is the opposite correct? Well, not yet. We can have the same height, but the people will be different. What we can though state, and that's why invariants are so useful, we can state that if invariants are different, if heights are different, then surely we are dealing with different people or different backbones in this case. So, and examples are chain length is a rigid invariant. And also, if you take total sum of distances between, for example, carbon atoms, those are invariants, which means that will be the same if we are dealing with the same backbone. And how about atomic coordinates? Well, those are not a very good example. Yes, they are routinely used in representation of proteins, for example. Yeah, all the files, they consist of coordinates, but formally, they do not coincide more often than not, which means that are not invariants. And we see that coordinates are slightly different. Well, what can we say about the backbones? Not much so far. There is another invariant, which is very oftenly used, that is torsion angles. And torsion angles, basically, so if you have, that is the angles between the planes that are formed in the backbone. So if we take any 4 atoms in a backbone, we can consider 2 triangles that are formed by the first triple in those 4 atoms and a second triple in those 4 atoms. And we can measure the angle between the semiplanes that are formed by those triangles. This is the torsion angles. So here they're briefly represented, and quite often only 2 out of 3 possible torsion angles are considered, and indeed there are invariants, which means that if the backbone is the same, then the torsion angle's geometrical shape is the same, then the torsion angle should be the same. Again, they are not complete invariants because the opposite is not guaranteed to be correct. So why, again, they are not complete? Because, well, just by the degrees of freedom, just considering those 2 torsion angles will give us 2m-2 degrees of freedom, whilst to consider all possibilities by all possible things that are changeable in here, that is, that will give us 9m-6, and I will briefly come to that a bit later. Okay, so we had an implication in one direction, logical implication in one direction, if then, yeah? What if you want to have the double implication or reconstruct from invariants our backbones? In this case, mathematically, such invariant is called complete invariant, which means, well, as written here, the coincidence of invariants will imply the coincidence of the backbone structures. Invariants are interesting by itself, but we are dealing with quite flexible shapes, which means that it will be nice to measure not only how, if they are equivalent or not, but how similar or non-similar they are. And in mathematics, that is done in their distances or distance metrics. And for something function to be distance in mathematical sense, 3 axioms should be satisfied. Well, the first one is identity, the second one is symmetry. Well, basically, if you swap places for 2 objects, the distance should be the same. Quite obviously, yeah, we compare A and B, we compare B and A, we would like to have the same number. And the triangle inequality, again, that one is useful to run many clustering, for example, algorithms, especially efficiency and time. It is sort of embedded usually in the algorithm. Well, we have many different metrics without even realizing that we deal with them. For example, the trivial discontinuous metric, if you have a complete invariant, basically the binary yes/no answer is already an example of metric. So if something coincides, a metric can be defined as 0, and if something does not coincide, that distance can be defined as 1, and it is also a metric or a distance. So a distance is not only Euclidean distance, which is continuous as we usually measure, it can be lots of different things. Yeah, remember, mathematics is the art of. Another example of well-known metrics is root mean square deviation. So it is defined like root mean square deviation from some kind of number. I'm not talking much about that. That's quite a useful and interesting measure to measure distance between backbones. But suppose we have those distances, and then the question is, then what? We've measured lots of distances. Yeah, we've got all those numbers supposedly in the table, but what to do with them? And ideally, we would like to see them in some kind of a map to connect further with some kind of properties to see easy how to compare them, which is close, which is not close. And for that, just the numbers of distances are not enough yet. We need to define the invariants to be able to associate the points or positions on the in the whole rigid space of the backbones. Well, there are common scores that are used in proteins, but just briefly, those are not distances usually. There are rather similarities. So TM score is one of them. LGT is also not a distance metric. And I have a couple of student projects which has found out even quite a few symmetry violations, which was a bit surprising. Why is it important? As mentioned, for clustering methods, many clustering algorithms, they rely on having proper distances. Okay, so let's formulate our main problem that we are trying to actually solve. So our goal is to find such a map that maps backbones into the parameterized space in such a way that this map is an invariant, which is written here, and it is also a complete invariant. So here we now have if and only if, but which means that any backbone should be possible to be reconstructible from the invariant. Quite an interesting and unique property. Well, a bit similar to trying to reconstruct the human, not just from the height, but from something else like DNA, for example. Well, we also would like to represent them, compare them, because, yeah, rigid motion is good, but not enough. And comparing the backbone structures is essential, which means that we also would like to have a metric. And in this case, we would like this metric to be not a discrete one, but a proper continuous metric. And here I formulated the strongest type of continuity, which is well, Lipschitz continuity, which means that if the backbone structures are perturbed slightly, are not the shapes of them are not too far from each other, then we want the invariants also not to deviate too far. And inverse continuity also would like the opposite. Since we can reconstruct the backbone shapes from the invariant, it would be also very nice to be able to do this continuity in the other direction. That is, if we have the invariants that are quite close to each other, then we can match the backbones also quite well. And another very important practical requirement is that all this from 1 to 4 should be computable pretty quick. So that's the problem formulation. Let's move into the implementation, into our solution. So we introduce a so-called BRIE, backbone rigid invariant, which is defined in the following way. We start with a chain, and the chain proteins is good because it is has a unique order from the first element in the chain to the last one. It's not, the backbone is not a cycle, it's just a linear, well-ordered chain, as mentioned. Which means that we have the residue number 1, and in the residue number 1, we define an orthogonal basis, orthonormal basis. So remember there are 3 main atoms, N, A, and C. Nitrogen, carbon, alpha carbon and carbon. So we define the orthonormal basis based on this first triangle and the first residue of N, A, and C. And then what we do actually, we define the next residue in terms of the coordinates of the previous residue. So what is interesting in this case? The first idea was, well, if we define the basis for the first residue, why not define the whole chain just in terms of these coordinates? But then we risk sort of getting lots of errors and deviations, so that's not ideal. Whilst if we measure the next residue in terms of the nearest previous residue, then this error is minimized, but that also allows us to measure the sort of shape of the whole structure locally. And our theorem that we've proved states that this backbone rigid invariant indeed solves the mapping problem that I've just formulated step by step. So let's demonstrate how it does. So firstly, backbones are matched by rigid motion if and only if those BRIEs are complete invariants and they match. Why is it important? The Marjorie Seneschal mentioned on Monday, the paper that defined periodic structure of crystals as a rigid class of periodic point sets. And here, the same is done to the backbone structure. So backbone structure is also defined as a rigid class of all backbones by this BRIE invariant. It is also interesting that theoretically we did not do it yet, but we plan, and it is possible to extend BRIE into side chains as well, just by representing not only the major atoms in a backbone, but representing other atoms in the side chains in a similar way. So, well, so far we had our hands full with the backbone. Now, measuring what is similar, what is different, we can match the shapes, but it's not so interesting for the proteins that are inherently flexible. It's interesting indeed to compare them and compare them continuously. That's why it's interesting to have a distance, and we used the L infinity between the invariants to compare the backbones so far. And the beauty is that indeed everything is computable in a good time, in a linear time by the number of residues. So it is computable. So what happens? The BRIE consists of the rows representing the next residue coordinate in basis of the previous coordinates. Which means that if we had m residues, then 3 atoms of the next residue will have 3 coordinates each. Which means that by construction, B is represented by M times 9 matrix, just the set of numbers, while the first row is slightly unique. And those numbers have their just coordinates, which means that we can just flatten them to a vector of 9m minus 6 coordinates, 9m of minus 6 numbers of the same dimension. Why is it unusual? I will mention in a bit. So basically, we have the set of similarly behaving numbers, which means that we can now compare them by many distances that we have, including, for example, L infinity, which just compares the maximum deviation, or finds the maximum deviation of the coordinates of backbones, but there are other possible distances available, and I think the next talk will mention some of them as well. So Brie is also interesting because, well, I've started with a fixed length, but actually Brie also respects the variety of lengths in a sense that it respects subchains, which means that if we take any kind of subchain, then it's easy to compare them and reconstruct from the longer or shorter chain. So suppose we have a Q as a subchain with a smaller number of residues than initial M. If the start of 2 chains was the same, then we just sort of cut the largest matrix up to the number of rows that we need, up to the size that we need. If we started not from the first residue, then only the first row will be not as usual. It's the first unusual orthonormal basis of the first residues, but all other residues already should coincide. And actually, the first unusual row in the shortest matrix is also reconstructible from the coordinates of the next residue. Okay. Well, another interesting property is that it's very easy to distinguish mirror images. Well, it can be useful because, well, at least in some situations, I think, having mirrorly imaged proteins, I don't know how common it is, but they can have strikingly different properties. And having those kind of descriptions in Brie invariant, it's very easy to see quickly that those are indeed mirror images of each other because, well, just the sign in one of the column, well, in 3 of the columns, Z coordinates will be opposite. So it's easy again to see from the Brie. Okay, promised continuity. Yeah, here's the precise formulation of what it means. So let me remind that we are considering L infinity here. If we start from very similar backbones Q and S, then invariants will be also quite similar. And we can even compute how similar. So we can have some guarantees that the computed invariants will not go far from each other, that will also, the similarity will be preserved. And also, well, the constant of this guarantee can be computed by a formula. Actually, this formula has an interesting geometric meaning. It can be represented in terms of bond lengths, well, the range of bond lengths, and also the heights of the backbone triangle of main 3 atoms. And that probably sort of is related to the common assumption that bond lengths can be taken the same, and well, some other parameters can be taken the same, but now we can sort of really measure this common assumption as well. as measure its violations, which will be again the topic of the next talk by Will. So we have continuous invariance, and again remind that we have complete invariance, which means that when we have bijection, which means that yes, we are very much interested in reverse continuity as well, which means that if the invariants are close, then again we can find the rigid motion that will almost match the backbones. And again, it's represented in the terms of Lipschitz constant. And here, well, this constant looks ugly, but the next paragraph is just about how to make this mu less ugly, and after some renormalization, turn mu into just square root of 3. Well, basically, it's because of the diagonal of the cube of the perturbations. Okay, so let's compare to the classic approach. In classic approach, there is another length angle invariant. And I mentioned that the torsion angles themselves are not a complete invariant. But indeed, if you have all set of possible rotations in length, then it will be complete invariant. So by rotations in length, what do I mean? Well, firstly, there are 3 torsion angles, the angles between the planes. Secondly, there are 3 bond angles, the angles between the vectors in the backbone. And thirdly, there are 3 bond lengths. So if we take all of them into account, then yes, it will be indeed a complete invariant. Quite often, they are assumed to be constant, and as mentioned, there could be at least some justification behind it, but well, it should be probably spelled out a bit more clearly, and we have tools to do that. Again, hopefully we'll mention. So this length, traditional length angle invariant, we call it lay. It's a good invariant. It's a good invariance that allows to see how the shapes transform by changing the angles, rotating them. So in this sense, it's understandable why it was introduced. But length angle classical invariant is a combination of lengths and angles, which means that by definition the units are different, which means that well, quite a bit of work is required to compare them via any measures and means and any kind of distances. Well, angles are commonly computed with non-trivial additional functions, which obviously accumulate errors again in computations. While BRI is not like that, it's just vectors. It uses linear algebra, which means that no such complicated non-binary functions are involved. For example, continuity and the Lipschitz concept was relatively easy to complicate for BRI. For lay, it should be possible, but highly non-trivial. And yeah, it's easier to do it this way from BRI going to lay rather than other way around. But yes, we will conform the duplicates. I'm running ahead of myself. If L infinity is the same or is equal to 0 in BRI, it is equal to 0 on lay as well. So there are quite a few of similarities between two invariants, two sets of complete invariants, but computational speed is and computational easiness is quite strikingly different. Okay, so we are continuing the direction of cleaning the databases. So let's try to actually apply our invariants to the PDB dataset. And the first step was actually to take the PDB dataset and try to clean it just a bit further. Yeah, PDB do make quite a beautiful work and a job of cleaning the newly suggested entries, but still, well, nothing is perfect in this world. So we've started from 2020 the entries, and since in each entry there are more chains, we started with more than 1 million chains, and then we've ended up with approximately 700,000 chains after applying those steps. Some of them really would mean that probably we are dealing with something strange. For example, if the atoms in a backbone have very small distances or very far distances, then something strange is going on. But we also sort of removed disordered chains with which allowed various occupancies. Well, just for now, because we want to end up with a very refined structure without much doubt. And well, yeah, we also removed non-started amino acids. And well, let's make pairwise comparisons between them. So we've computed invariants for all the remaining chains, and it was fast. It was just in about 3.5 hours. And comparisons after that, because the BRI has the same essential dimension, then the comparison using common computer science algorithms like KD3, it was again fast. So overall, it was 6 hours. So what have we found? We found that more than 13,000 pairs of backbones have exactly the same zero distance L infinity, which means that there is, well, L infinity again is the maximum difference, which means that the difference between maximum difference is 0. Which means that those backbones can be exactly perfectly matched by a rigid motion. So that is our explanation of what it means that they have exactly the same shape. Well, that can be perfectly fine, because they indeed can be and are expected to have the same shape, especially, for example, if they're coming from the same PDB entry, yeah, and there are multiple shapes that are slightly, combined by some kind of rotation around each other, and they interact. So they may not be identical coordinate-wise, and that's fine. So backbones can have the same rigid motion shape, but coordinates does not have to coincide. But when we look further into it, we've discovered that there are quite a few pairs that have exactly identical coordinates themselves up to the last digit. And this comparison of the coordinates one by one was obviously not possible on the whole PDB data set as such, but after filtering them out using the invariants, then comparing the similar structure coordinate by coordinate became again computationally possible. And 700 of such pairs are also in different PDB entries. Well, as John mentioned, that's useful to measure how similar the structures are and if the accuracy or precision is the same. And here we all aim for a very good precision. But if we have the perfect precision, is it always reasonable? I don't know. In some cases it is, but in some cases, well, not really. And those are a couple of examples where there are totally identical carbon atoms with even different sequences. And at least one of them was accepted as an error, and we were praised for being able to actually find this error. Well, it was submitted by mistake. So when a new protein is deposited, it is validated individually, but we think that it's very useful to have actual comparison with everything that was deposited before. So we have here some, well, good quality structures that have still quite a few of with different amino acids, but exactly the same coordinates. For example, this is one of the examples. As you can see, one of the atom is changed, but one of the residues is changed, but the atoms are totally the same. Well, and well, there's distribution of the situation. So if we have identical primary sequences which are green, then we can sort of expect this situation. So again, total coincidence of the coordinates can raise question, but okay. But if the primary sequences are different, but we are still getting this perfect or nearly perfect precision, here's the perfect precision, here's nearly perfect precision, yeah, well, then from the sort of probabilistic point of view, I would like to understand what on earth is going on. Why is it happening? Is it a refining program? Is it something else going on? So basically, why? Is it some law of nature that we are seeing in front of our eyes? I don't know. So all in all, we've discovered a way, an algorithm to quickly look at quite a large PDB data set and compare all the structures within quite a short time just on a model's laptop, which can be especially useful with the current tools like, well, AlphaFold, and in crystallography, I think it's even more prominent when machine learning methods, they quickly produce lots of new entities or new measurements. And then the question is, well, are those measurements even different from each other within the same prediction data set? Because, yeah, machine learning tools are not producing just one, usually, suggestion. It can produce many predictions. So that's useful. That's why it's so useful to be able to compare them quickly to the whole variety of other data sets that we have in at our hands. Well, and yes, it's very important to use distances rather than similarities. Otherwise, if you fail triangular inequality, only any kind of clustering becomes just a bit questionable. And our next project with the students, hopefully, will be try AlphaFold again, but with our new inventions. Okay, but that's all for today. Thank you very much. Any questions?
Thank you, Olga. Please thank Olga for the presentation. As usual, let me stop the recording and